Good afternoon, students. We'll start our second lecture for the day. We have with us Dr. Ajay Kumar Mahato, Staff Scientist 3 from Center for cDNA Fingerprinting and Diagnostics, Hyderabad. He has graduated with a PhD in Bioinformatics from Dr. APJ Abdul Kalam Technical University, Lucknow. He has also a master's degree in Bioinformatics from Chaudhary Charan Singh University, Meerut. He has been working with as a research associate with the National Research Center for Plant Technology, New Delhi from 2006 to 2021. He joined in 2021 at the Center for DNA Fingerprinting and Diagnostics as a staff scientist and group head. His major research, he has 13 years of research experience in the field of bioinformatics, especially in genome assembly of Sanger, NGS, and SMRT sequencing data. He has worked in 2 international consortium projects from India sites in like International Wheat Genome Sequencing Consortium and SOL Genomics Network, and has worked as a lead bioinformatician to assemble wheat and tomato genome. Apart from that, Dr. Ajay has assembled more than 5 graft level genomes for mango, PGMP, jute, coconut, magna pote oresa, and 2 reference level mango and PGMP genomes, and performed further annotations and other downstream analysis for which he has published several peer-reviewed journals. He has also experiences in high throughput genotyping to QTL identification and mapping, diversity, phylogenetic studies, GWAS, linkage map development, fishing of genome assembly, and other analysis. He has more than 44 publications in international and national level peer journals, and also has authored 2 book chapters, has provided training to various organizations, and has developed 3 designed and developed 3 gene-based SNP Affymetrix array genotyping chips for PGMP genome, mango genome, and Amaranthus genome. So with this short introduction, I invite Dr. Ajay to deliver the lecture. The floor is yours.
Thank you so much, Doctor. And I would like to thank Srishti to take this initiative along with BIIS, so which helps student to deciding their research domain, or the budding scientists who are willing to take this science as a career. So traditionally, the science is basically, everyone is talking about, okay, it's a purely wet lab in which you have to add some chemicals or like extract the DNA, all those things. But as the era has been changed, now this science is also getting changed, right? So there is the many things which is now got automated, and the data generation is very huge. So as you introduced me, like I have like experience in data analysis, or you can say data science, especially in genomics. So, and the future is genomics, right? Because of this bioinformatics approaches and the genomics approaches, we are able to deal in a very rapid and a very strong manner in this pandemic against that coronavirus. So in this my talk, I'm going to just briefly discuss about like what is bioinformatics and how students like the graduate and the postgraduate, they can take it as a forward as a career. Okay, because it has an ample of opportunities for everyone, for a wet lab scientist also, for a B.Tech student, for M.Tech student, for MSc student, for BSc student. So keeping that aim as a main thing, I will just start my presentation. Let me share my screen first, so that yeah. So I think it is visible to everyone.
Yes, Doctor.
Okay, and I request all the students, if they have any question, they can interrupt me in the middle, and then get that rectified. Okay, I just want to make this talk like not a monotonous, but it's a bi-directional. Rather than taking, I will take the question at the end. But if you have any doubt in the mind, just talk me, just stop me and just ask that instantly. Okay. So let me write this. Right. So I will, what I will do, I will just stop my video so that the bandwidth will be less utilized. I think now, so my slides are visible.
Yes, Doctor.
Okay. Okay. So thank you so much again, and I'm going to deliver a talk, or you can say a lecture on this fundamental of bioinformatics as a tool and databases. So myself is Ajay Kumar Mato. I am a group leader at Center for DNA Fingerprinting and Diagnostic, and I am heading my own lab called the Lab of Genome Informatics. It's a completely in silico lab which do the research in the area of human genetics, human genomics, as well plant genomics, plus also like development of softwares and new algorithm. So the outline of my talk is like, I'm going to talk brief about what is bioinformatics, and what the word bioinformatics means, then followed by like the how this bioinformatics concept come, like basic history of bioinformatics, how a bioinformatics can be used as a technique to find the answer of many complex problem which is not possible when alone. So, and then areas in bioinformatics means what are the application area where these days in this coming in this time and the future, it is widely used, or going to be used, and what are the main application of bioinformatics. So in which I will talk like bioinformatics is everywhere in the science, whether it's a domain of you can say plant pathology, or you can say like microbiology, or you can say botany or zoology, or molecular biology. Everywhere the bioinformatics has a huge application. And finally, what are the myth and the facts about bioinformatics? Because there are many myth and facts which need to be clear to the student, so which will help them to decide whether they can take bioinformatics as a research career for further in their future or not. So can anybody tell like what is bioinformatics? Hello?
Sir, a blend of biology and computers.
Yeah, that's very great. So bioinformatics, as you see, it's a bio plus informatics. So it's a area where this biology and information science, or you can say information technology, merges to form a single discipline. So in this, let me pointer. Right. So in this diagram, you can see, so this is bioinformatics, which is the amalgamation of computer science, chemistry, statistics, mathematics, biology, and biochemistry. So you're right, the bioinformatics is a science where the biology and IT merge to form a single discipline. So this definition is I have taken from NCBI, which is the world biggest database related to bioinformatics and other resources. So in a layman language, we can say like bioinformatics is a marriage between a computer science and a molecular biology. So how a computer science means, a computer scientist is basically work on the algorithm, and based on the algorithm they design a techniques, right, to solve a problem which is faced by a molecular biologist, right? So let's say I'll give you one example. So the so there are total 10 gene in the coronavirus genome, right? But how they have identified the genes, it is based on the algorithm which find the start and stop codon. So that is making use of the software, and software are based on the algorithms, right? So so this IT, information technology, is very much used in these days in all branches of science, and also for the analysis of biological data. And based on the new algorithms, they design and develop new tools which will further ease the life of the researcher to solve a complex problem. So what are the applications of bioinformatics? So you can see in this image. So bioinformatics is a building bridge which bridges all the science, probably like animal plant, computer science, statistics, mathematics, biochemistry, and the evolutionary science, everything. So bioinformatics is a building bridge which so let's say if you have a sequence, I'll give you one example. So let's say I have a plant pathogen sequence along with me. So by using bioinformatics or computer science, okay, and mathematical tools and statistics, I can get their basic statistics, like how many gene it is having, like whether it is rich in what is its base composition, how many genes, what are those genes perform, whether those genes perform which function. Okay, now whether out of that gene, how many are like pathogen causing, and let's say if that pathogen, how evolutionary it related to other relatives and other species. So bioinformatics, based on this single thing, using all these domains and the softwares or the ideas, we can draw a conclusive conclusion about that sequence or about that new pathogen. So this is a cartoon. You can see somebody tells, okay, it is used for the drug designing, because any earlier it is highly used for the discovery of lead molecule, which will further translate as a potential drug, right? And basically somebody, it is a genome analysis. So now after this coming to the new generation of sequencing, next generation you can say, where the throughput of sequencing is high, genomics is like major changes in genomics and its application has started. So mainly people think it's a genome analysis, like and due to this genome sequence or the sequence availability, the evolutionary studies based on the sequences is rapidly increasing. Why? Because the computer scientists, they have developed many tools and software, and might be certain tools you student has used also, like a multiple sequence alignment in MSA, cluster W, cluster X. Okay, now for a mathematician, it's a basically algorithm development to solve a complex problem. And for statistics, it is statistics. For a statistician, it's all about the data science, right? And for programmer, it is bioinformatics is like maintaining all the data in the form of database. So it's a huge application, you can say. So for a biologist, it's a basically biologists collect basically the data, where data could be anything. It's a sequence or a microarray data or any phenotypic data. And this computer scientist, or you can say computer science or information technology, along with mathematician, statistician, they develop new algorithm and tools and softwares to analyze this new type of data, or to analyze this data in a new angle, right? So that it will conclude, it will give you a new clue about the data which is still unknown. So you can say bioinformatician study biological questions by analyzing the data using the software which was developed by the computer scientist, and come up with a result which is statistically higher value, and it has a conclusive uh, conclusion, so that a biologist can take a further decision. Now, there is huge confusion, like bioinformatics is a technology or is a methodology. So, can anybody tell like bioinformatics is what? It's a technology or a methodology? Student, what do you think about like bioinformatics? It's a technology or it's a methodology?
Sir, I feel it's a methodology. I mean, because it's like building upon something that's already existed. So you make some pipelines and all those things.
Okay, and why not? And so according to you, bioinformatics is a methodology, right? And why not the technology? You know, actually, if we see bioinformatics is both, it's act as a technology as well as the methodology also. You know, what is technology? So the technology is like technology means you are it when you are managing these things. You are making the query of that thing and analyzing the data. So let's say in as a technology, I'll give you one example. So it's a bioinformatics is a very much powerful technology to manage, right? Big data like these days, human genome, plant genome. That like India has started thousand genome sequencing, in which the ICMR has to sequence 1,000 genome of the diverse population across, counting from this Kanyakumari to the Kashmir, from Leh Ladakh to Gujarat. Okay, so it's a powerful technology. So the data generation is in petabytes. Like you can see petabytes. It's a huge data. So as a technology, bioinformatics is now, there are many positions which are coming, which only deals with the management of data, so that the data can be stored in a structured manner where user can make a query and fetch the information from that big huge database, right? And as a methodology, as you told, that like it's a top down approach. Whatever software is available, you are using it for the analysis of XYZ data to find new patterns, or discovery of new element, function element, or like new validating new hypothesis. So bioinformatics is act as a technology as well as methodology. So student, just remember this thing. So here this act as a tool, plus it act as a database also. So the database itself comes like a technology. So when I call a database, so there are concept of these data lakes. Probably the computer science students are aware. Earlier the database which is smaller, now the concept coming like a data lake. Okay, and data warehouse, where the huge the data storage is much higher than the a database. So this is an example I will give you as a technology, how a bioinformatics you can use. So let's say genotype and phenotype. Can anyone tell me what this word genotype means and what the word phenotype means? Student?
Genotype is the genetic information that is present, whereas phenotype is what we see externally, like what it presents as.
Okay, so means genotype you cannot see, but phenotype you can see, right?
Essentially, like on at a surface level, phenotype is what we see. Yes.
Yeah. So it means like you are absolutely correct. Like genotype, it's at the genome level, and phenotype at the like surface level, I can say. So just a careful look into this example. So when we talk about genotype, so let's say I'll give you one example. So one DNA sequence from a X genome is given, right? Or a genome, new genome is decoded, or its sequence is given to a student. So what things he can perform at this point? Like from that sequence, he can find like whether it contains any genes or not, right? And he or she can perform the comparative genomics analysis using the BLAST. And what is comparative genomics means? Let's say you have a piece of DNA or a sequence, then you are just finding like from what other organism it is showing similarity. So that is comparative genomics. And then based on that, you can see the evolutionary pattern of the provided sequence, how that sequence evolved. So from DNA sequence, right, you can predict the gene, you can do the annotation, you can make a comparative genomics, and you can make a evolutionary conclusion by designing a or generating a phylogenetic tree or dendrogram. Okay. Now let's say from DNA it goes to RNA, right? So at the RNA as a technology, this bioinformatics you can use for like motif finding. So using the software. So because like in RNA, this motif is important. Motif and domain are the main part of RNA, or you can say a gene, which governs the function, right? And these days, now the RNA is widely used for the identification of the expression of certain gene at different condition. So I will give you one example, right? So I have a rice plant, okay, of let's say basmati, and I grow it in normal condition. Now the same basmati, same breed, I will give the stress of salt stress, right? And after I observe it, like after 15 days, this the salt the sample which grown in salt, so its growth is stunted. Now as the variety are the same, the seeds are the same variety, but why there is a stunted growth in the saline soil? So everybody know like salinity is a main factor, but it is this stunted growth is only governed by the few expression of genes. So when you extract the RNA of both the plants and do this RNA sequencing, which is very common these days to find out so you are able to find out like differential gene expression pattern. Like let's say one X gene in a normal condition, it is highly regulated, but under stress condition, it regulation goes down. That may cause like this stunted growth, or a Y gene which is like very low expressed in the normal condition, but under this stress condition its expression goes exponentially high, which hinders the biological pathway and causes the stunted growth. So on the RNA on the RNA data as a technology, you can use like this. From the protein, like everyday like mass spec data is very much common, which is used for the structure prediction and structure alignment. So you can see like whether your mass spec data is from which component. And molecular network like protein interaction, you also can identify using this bioinformatics as a technology, right? So you can easily pinpoint, so if that protein interaction is happen, like how it hinders the signaling network on the regulation of the network pathways, right? And cell like you can say like from cell like population genetics or human genetics. So in population genetics, it is let's say cells may, if you are using with dealing with the SNP, you can easily find out like a same gene which is present in two different human you can say two different person. One is a normal and one is having a disease. but why, but that is only there is a SNPs or the base change in the gene of that deceased person genome, right? So that is color blindness, all those things are the example of this. So this is how you can use bioinformatics as a technology. Now approach it. So let's say a I've come up with a biological question. So the question is whether the gene regulate by a protein X or not. So for that I have generated a data like chipsec data, and now I have translated the data into, feed it into a computer program of high-end computer program like HPC to solve the task. And what is the task? That align the read and do the clustering. Okay. Now this now let's say it is not solving my problem. So bioinformatics develop an bioinformatician develop an algorithm and then this data set is chosen for its standardization. Now the implementation, like then I implement the algorithm as a software and run that application to align the read, predict the gene, and then give the conclusive result, right? So the finally it will answer the biological question in the form of whether this reported protein regulate the X protein or not, or whether its effect is binding sites or not. So this is an example of a whole process as a technology, and as a methodology, as the student tell, like everything is available on the is available in the public domain, like the softwares and the databases. Now the only as a methodology you have to take the data and just do the analysis. So let's say for the data management, like if you are willing to manage develop a database, you will go for the data management, which where it store the data in the form of metadata. If you want to get the analysis using softwares and very now these is huge web servers are available. Okay. Now for the data mining, for the data discovery and simulation modeling. So the proteomics, right, in which the simulation and modeling is a very important. So by this way, you can use the ready-made software for analysis. It's called methodology. It act as a methodology, but when you have X problem and you are not able to solve by using the existing tool, then a bioinformatician will develop a new algorithm and translate into new software to analyze the data, and so that it will give a conclusive result. So by this way, it is act as a technology. So I hope like you now the students are clear about this. So bioinformatics is not a methodology; it's a technology also. Day by day, every day you will get like every week there are new softwares are coming up. Okay, and why? Because these is like most of the this biological student now they are training themselves in the programming part, like Python programming, which is mainly used in the field of bioinformatics, shell programming, and they design and develop new algorithm to solve the problem, and or to draw to see the data at the deeper level. So the first protein reported was a bovine insulin in 1956, and after nearly a decade, so the first nucleotide acid sequences of a yeast, which is 77 base pair, was you can say sequence or decoded. Now this 1965, right, the day of gathered all the available sequence and created the first database, which is called Atlas of Protein Sequence and Structure. So this is the world first reported database called the Atlas of Protein Sequence and Structure. Now these days it is now becoming from Swiss Prot. You all students are aware about it. So the initially the original Atlas of Protein Sequence, now in 1972, it takes more data of crystallography and protein structure, then the protein data bank, and finally now 1987 onward, it is known as Swiss Prot. So in this slide, you can see this is the history of bioinformatics. So let me just go ahead. Like in 1945, you can see the first computer evolved. In 53, a double helix DNA structure was reported. Now the first in 54 also first protein sequence was solved. Now in 1960, you can see this molecular evolution clock was developed, right? And 79. So this is an important time. Now in this 69, that APRANET is created. So APRANET is created by the US Army to communicate within themselves during the warfare. And from ARPANET, this internet is evolved. Okay, it provides feature of email, Ethernet, and TCP protocol. And during the year of 77, the Sanger sequencing, like dideoxy sequencing is discovered. And now this technology is goes higher. Now 1980, 82, this PC, or you can say personal computer, was launched, and the internet is coming into picture, as like they are able to communicate across the other country also by using like DNS approaches. And in 1990, this Human Genome Project was started. This Human Genome Project, it was completely based on the Sanger sequencing data. So you see, in 1977, the Sanger sequencing was discovered, and in 1990, this big Human Genome Project was started. And the aim is what? The aim is to decode entire human genome, or to read the whole human genome, and and its basis like IBDC, and find out like how many genes will present and what are those gene function, right? But the approach is based on the Sanger back by back sequencing approach. So it is very time taking process. It include, the first one is generation of physical map using the human genome DNA. Then each back is used for go for cloning. Then it will go for the sequencing. Then sequencing, like and the library size is varies from 120 to 130 KB. From sequencing, it will go to this bioinformatics part. And those days, I'm talking about 1995 or 1996, or so, that it is the software are very meager. Okay, but and most of the software, scientific software I'm talking about, they are based on the Linux. And so this software called FredFrap and Concept, was used for this human genome decoding. And luckily, several other genome sequencing projects are going on. In 1995, this human first influenza genome was sequenced, followed by the yeast, earthworm, and fly genome. And 2005, and then 2001 to 2004, this human genome was sequenced, and it was released. And recently, like last year, in this year only, in the month of February, the updated version from like telomere to telomere sequence of human genome was released. And due to this genome sequence availability, there is a huge, you can say, huge scientific development, and the cure of disease came, right? In terms of vaccine, new drugs, like personalized drug, everything. And 2015, this next generation sequencing was introduced. And this is the era from where this genomics has taken speeded up. And now this everywhere you're talking about genomics and genomics, like genome sequencing. So, and I request all the students to just visit one website. I forget to put the slide, called the Earth Biogenome Project. So it's that project, you can say it is going to be, the aim is to decode or sequence whole eukaryotic genome, which was whole eukaryotic species, whether it's a plant, animal, bacteria, fungi, virus, which is available on the earth. Okay, so you can say it's a like a event which is you can say highly impact after the landing of human on the moon. So this, let's say, and in terms of now that is a history. Now, if specifically if I talk about the bioinformatics, so the for 1552, the computer science, this computer program of protein structure was developed. Now in 62, this Dayhoff has come up with the first protein sequence, followed by like the 71, the word bioinformatics came into picture, and followed by like the generation of matrix PAM 1978 PAM, that is per percent accept mutation matrix, which is widely used for the phylogenetic analysis and BLAST algorithm. 1990, this BLAST come into the picture, which has changed the scenario of comparative genomics. Earlier, the people are using dot plot to seek or to compare two sequences now, but by the introduction of BLAST, that's their life become very much automated, and it can able to align two sequences up to any of the length. So this is also a technology platform in terms of if I talk about like when this project was started, and majorly this is related to the sequencing part. Like in 1997, you say Sanger sequencing started first, right? Which is followed by like this several genome sequencing. The major changes come here in 2005 and 6, where the this next generation, or you can say the Pyrus sequencing, come into the picture, which includes Illumina and Selexa, and which has started like given a platform for the scientific community to start the genome sequencing project, like 1,000 genome sequencing project, like 3,000 rice genome sequencing project. And this technology is getting so advanced. Earlier, the getting the sequence is very much costly, but now the sequencing is very much cheaper, and the data throughput is very high. These days, this Illumina NovaSeq, in single run, student, can you imagine, in single run, it is generating 6 to 8 terabyte of data. Can you imagine TB in terms of so if I'm talking about TB, this is in terms of file size. So a normal hard drive, like you can say, so 6 to 8 TB of data is generated per flow cell. Now for analysis, you need high computational power and bioinformatics expertise, or even you are a student of computer or a biology, you should have some Linux or IT exposure to analyze or handle the data. So that is the main reason. If you just after this session, if you Google it, like the job opportunities for let's say for a biotechnologist and for a bioinformatician, you will see a huge difference. Now these days data is everywhere, but there is a scarcity of the biologists who understand the data on the concept of informatics. So, and the future is going to be like I can say like 10 to 15 years from now onward, it is going only for the genomics and the data science. And the data science is not is what like this data analysis, but also this this artificial intelligence. So I have started working, my lab is working in the area of artificial intelligence, deep learning in data science, where we are just making models, okay, to predict like the probability of getting the disease of that rare disease in that particular patient. So this data science, or you can say this informatics, is going to be a future. So can anybody tell like what is the main ingredient of a bioinformatics? Student, can you guess?
Computers or data, algorithms.
Yeah, absolutely correct. So the main ingredient, as she told, like data, and even more data. So the main ingredient is like data. If you have a huge data, then this informatics come into picture and make the scientist or a researcher life easy. For analysis part, if that data is complex, new algorithm can be designed to explore the data. So so if I define a data, so as per the Wikipedia, right? So data could be anything, any fact, any statistics, any information, often numeric, is called a data. Okay, so the data could be scientific data, financial data, or meteorological or natural data like phenotypic data, transport data, any data or any reading or any information which you can record is a data for a bioinformatics application, right? That could be, it's a phenotypic, like a long leaf or short leaf. Okay, the diameter, so it become a data. Genomics, its sequence become data. Cluster, so let's say it's zero, it's if that growing cluster, or it is widely used in any cultural thing, or a disease which is prominent in only one type of that community. So this is all like data. So in the layman language, it's our individual facts, statistics, or information which is often numeric, called the data. And so can anybody, everybody is aware of like database or data bank, right? Like most of the students, like they are just using Google, right? Using this Ghana app and all those things. So can anybody tell like what is a database? Because in daily life, everyone is dealing with a database. Every minute you are dealing with a database, or even every second you are dealing with a database or a data bank. Please.
But it's like a collection of a particular thing. Like if you're looking for maybe Indian plants, so you can have a database for it. Or for just the proteins, you can have a database for it.
Yes, right. So you are absolutely correct, right? A database is basically a place where the data, similar kind of data is collected, it curated, and it is kept for the use of uh, public, or you can say in terms of science, like other researchers. Initially this database is not a so this is not the word which is widely used. So there are huge discrepancy. In UK, everybody say it's a data bank. In US, they are telling no, it's a database. So the solution is that the consortium come into the conclusion that okay, we will take the abbreviation as a DB. Okay, so database is basically DB. It's a short form. So a database is a collection of related data that you told is absolutely correct, and it is archived by using computer in an organized fashion, so that the information from there can be retrieved easily by the user. Okay, and second, and the data which is present, it is interrelated. Okay, and the database should be free from its any data redundancy, means duplicate data is not supposed to be present in a database. Okay, so because if it is there, it become non-functional. And these days integrated, or you can say interrelated database are very common, right? So I will give you one example, right? So initially I have created a database of my city, of all the like the states, all the states of the India, right? Now it is only its name. So anybody can and when anybody put its name, so it will tell it's their geographical location, like the coordinates from the Google map. Now I have another database which contain, like let's say a state called Gujarat. How many districts present in Gujarat, and what are their location? Okay. Similarly for all the 31 state. Now these are two independent database, but if we integrate these two into a single database, then it become more informative. So let's say if I search, when I integrate both the database and then I search for Gujarat, okay, Gujarat, okay, in Gujarat its coordinate is like this, and it is having this much number of district, and each district have like this much number of talukas. So by this way, this database, integrated database, is very important. And these days, everyone, like everywhere, you are getting the integrated database, whether you are searching via Google, or you are listening to your Ghana app, or any app like WhatsApp, or everybody is familiar about WhatsApp. So if you see in your phone memory, WhatsApp itself create a different databases, like one folder for image, one folder for video, one folder for message, right? But all those things are integrated with each other, and it will give you a single front page to you, which in the form of you can say WhatsApp, in which WhatsApp you can query na whether Ramesh has sent me message, okay, when this Ramesh has sent. So in WhatsApp we can do the searching also na. So retrieving or calling or firing a query is called search in a database. So in brief, a database is a collection of data which is structured, which is in index or it's searchable, and which periodically updated, and it should have a cross reference, means it should link to other database, right? And it also include associated with certain tools, right, which will help the user in the retrieval of information, or updating or accessing that database, right? And the data in the database, the data storage management is basically in flat file format, and the relational table was generated to link two or more the database tables or a database to form a bigger database. So now this is the concept of database. Like a database is simple, a collection of related information or structured way in a digital format, right? And but what is a biological database and why it is needed, right? So how many of you have accessed any biological database? Can you name any of them, student?
IBDC.
NCBI, anybody access DDBJ or Swiss Prot? Anything you have accessed over the net? Okay, yeah. So basically, biological database demand come after this technology advancement, right? After 2000 or after 1998. So where this you can say that microarrays become it is popular during that days, 19 into 2000 to 2009 or 10. It's very popular to see the differential expression, and it generate huge data. So for storing and just communicating the data, large scientific data, this need of biological database come into picture. Okay, so that it will be available to the scientific, not the individual scientists, but it is available to the whole scientific community via internet. Okay, and the data is always in a human readable form. Now the different classification of data is called primary, secondary, and aggregated. I told, like, the primary means I did some experiment. So let's say I sequence my genome or a new coronavirus genome, and directly store that result into the database, which is called primary database. Now, my another colleague have used the data, you have just query the data and get the sequence and perform the gene prediction, its annotation, and also he performed the phylogenetic analysis. So that he come to know, okay, there are 10 or 11 gene in that coronavirus, and there are mutations, and due to this mutation it become whether more pathogenic or less pathogenic. So this information he kept into another into its own database. Okay, so which you can say like secondary database. And when and the third person, he link both this primary and secondary to form an aggregated database. So the primary database is contain only the raw information. The secondary database always contain the filtered and useful information, while the aggregated database is amalgamation of primary and secondary. So let's say if you see the X gene in that coronavirus, whether like what is its exact location. So if you go to the primary database, you will get the exact location, right? So by this way, there are three classification of biological database: primary, secondary, and aggregated. So there are different based on the availability. You can classify a database into several category, like publicly available, which is with no restriction, like NCBI, UniProt, right? Or you can say EMBL in the scientific term. Okay, and there are second thing like available but with copyright. So you can say Aadhaar database. When you are getting your Aadhaar in a soft copy from the Aadhaar website, you have to give authenticated. Okay, and it was a and it was copyrighted to the government of India. The second one is accessible but not downloadable. So you can only view, but you cannot able to download. Now there are academic databases which are available, but they are not free freely available. So you have to create a login, and that admin or the user, that database administrator has to approve your login. Then only you have the access. Okay, like there are n number of database. I tell you, Giri is a database which contain the repeat regions of various from human to pathogens. Now, and the proprietary database, like the commercial availability database, you can say, for academic also, like Baiju and all those things. So those are like proprietary databases. Now, based on the type of data in scientific term, there are nucleotide sequence, there are special database for protein sequence, special database, it's for base, which is based on the its motif and 3D structure. There's special database. So let's say nucleotide database is like NCBI, NR database, or DDBJ protein ka sequence like SwissProt, or protein sequence pattern motif like you can say InterProScan, also 3D structure, SwissProt, or PDB. Gene expression database like gene expression omnibus, metabolic pathways like KEGG, mass spectrometry database like DALI. So in this way, there are many categories of database. For life sciences, you can say this 7 and 8 are very important, like for DNA, protein, for genomics, 2D gel mass spectrometry, 3D structure, biography, bibliography, and microarray, and other. So I will give you an example of what is primary database, secondary, and tertiary. So any of you can tell me like which one is the Indian nucleotide database, first Indian nucleotide database, which is designed, developed, and hosted here in India, in the capital of our country, in the New Delhi. Can anyone tell its name? And it is highly advertised in newspaper also. No? So the name is Indian Biological Data Center. So this is going to be the NCBI of India. Okay, it is located in the New Delhi in the Regional Center of RCB, Regional Center for Biotechnology, which comes under Department of Biotechnology. Okay, and this is like this question come into the IAS exam also. So please update yourself. So like only NCBI and EMBL and GenBank is not. India has its own like biological data center where you can submit anything, right? Like NCBI, where you can submit protein, nucleotide structure, or phenotypic, genotypic, everything you can deposit here. Okay, to address the IPR or to intact this intellectual property right. Okay, so the primary database is just only contain the raw data which are produced and submitted by the researcher worldwide. So I tell you, like for the protein there are PIR, MIPS and other database. Now this slide is basically I have dedicated to the Indian Biological Data Center, right? So being an Indian, I feel proud and happy that okay, yes, Government of India under the leadership of our this ministry, they have taken it further and developed this first biological data center, which is going to be the NCBI of India, and very soon it is going to be popular like NCBI or DTBJ. So it's a country first national biological data center for sharing, annotating, and the biological data, and which is coming from the various funded project which was funded by the Government of India. So let's say agriculture, livestock, microbes, humans, everything, every data, whether it's a genotypic data, phenotypic data, everything can be will be going to be stored in IBDC. It will give you an accession like the NCBI is giving, and then you can use, and you should use, and you must use that accession for the publication. Okay, it's because it's a national proud, like okay, like anybody has if in the manuscript you have to give the location. So like the Chinese, they have their own database now. Every paper they are just quoting that accession. They're not submitting even the data to NCBI. Okay, so we have to also take this thing in our mind. Okay, IBDC has like 5 major things, like it accepts the databases, it do this quality control, curation and annotation, and it take the data backup, its management, and also it is highly involved in training and outreach activities. So let's say if your institute or your college want to get an update or what is IBDC, you just contact with them. They will give you an online session with you, and they will teach you how to submit the things here. Okay, it's a one point data access for data archive and data transfer, right? And like most of the government institution now, they are joined with the NKN network, National Knowledge Network. So which is a intranet, countrywide intranet. So you can easily, if your institution using NKN network, you can easily support and access the data on a higher speed, like 100 Gbps or more than that. Okay, and it enables like the science researcher to deposit data at a central repository, and so that it is publicly available for the research, and also for the other person to make further use of it. And that is about the India and the global diaspora. The NCBI is a major player, it is the first database, and it was developed by this US National Institute of Health, USA, and the search engine is like Intros. Most of you are aware about it. You can search for anything, genes, protein sequence, genome, or any literature or anything. Okay, even expression like the microarray also you can search here. This is the this URL for the NCBI. Now, okay, can how many of you are aware of NCBI? Can you please tell me, student? Hello, is anybody there? Just make a wild guess. So I say NCBI is a data bank. How many database it contains?
Around 50, sir.
50, right?
Yes, sir. I'm just guessing.
Okay. No, not till date. Like it contains total 39 databases, right? So NCBI is a data bank which manages this 39 databases, right? And it is a mission program of United States National Library of Medicine, which is a part of National Institute of Health, NIH, USA. Okay, so and this is the list for let's say for literature. DB, I told you earlier, na? So there is a clash like data, whether it's we call it data bank or database. So they come up with a conclusion like DB. So in literature section there are 5 databases: books, mesh, NLM catalog, PubMed, and PubMed Central. Most of you are aware of like PubMed Central, PubMed, and books, right? And in the gene section, like there are 7. In the health section, so there are total 39 database which is managed by the NCBI, where you can find everything related to the science. So this nucleotide sequence archive database, okay. So EMBL, DDBJ, and so the GenBank, ELBL, and DDBJ, they are the 3 primary nucleotide databases. And the interesting thing is that on a daily basis, all 3 databases exchange the data among themselves, so that every data replica is available at the 2 different locations. Okay, so let's say if I submit my data in the genome data in the GenBank NCBI, by next day it will replicate to DDBJ and EMBL. So I can find my data with the same accession on EMBL also and DDBJ also. Okay, now the third thing, IBDC is also doing the same thing. If you submit a data at IBDC, right, it will generate an accession, which and they are in talk to like every day sharing like these 3. So the same accession you can find on DDBJ. The same data is replicated on GenBank and EMBL. So this is the EMBL is a European molecular ecology laboratory in Heidelberg. It was created in 1980, and now it is maintained by the EBI Cambridge, and it also contains the same data set what the NCBI is containing, along with some new tools like MEME and other tools, which is available, which is not available anywhere. So this databases is not just offering you the data access, but also offering you several software tools to analyze your data. Now, DNA Bank of Japan, it was started by a National Institute of Genetics in Mishima, and they're still maintaining it is led by the Takashi Gojobori. So this was developed in 1984. Like most of the you might be not born by that time, right? So you can imagine that point of time, 1984, the technology is rapidly moving toward the data science, right? This bioinformatics is already into the picture in the form of storing and the providing the data accessibility to the scientific community. Now, protein sequence, if I talk about protein sequence, so as I told you previously, that Atlas of Protein Sequence is the first database you can say, okay, and by Margaret Dayhoff. So Margaret Dayhoff is the father of bioinformatics. This Margaret Dayhoff is the father of bioinformatics, and it was published by this Atlas of Protein Sequence and Structure. It was initially published in the form of book, right? We keep on publishing 1978, and then after it become PIR, and it become digitally accessible to everyone. So these are the list of protein databases. like SwissProt, Tremble, Interpro, and Go protein profiling and Inter. So these are the various databases which is related to protein. Okay, like Interpro is basically, if you go to this database, it will tell like if XYZ protein contain this 2 or 3 domains, and these domains perform this function. Okay, this Tremble, it's a translated sequence, nucleated sequence of EMBL. So whatever nucleated data present in at EMBL nucleated archive database, its translated version is protein. It is stored in TREMBL. Okay, and the Swiss part is the world biggest and the largest database which contain the annotated protein sequence. Okay. Similarly, from like protein profile, which contain the profiling of the protein. So by this way, there are n number of databases. This is specific for structure like MSD, 3D-Sec, DALI, D, and BD. And there are the list of like literature database, which I already discussed, right? PubMed is basically, it contain all the free or publicly available research article as a catalog. So rather than going searching on the Google, you can directly search on the PubMed using this you can say author or its title or its domain. So this is now the latest homepage of NCBI, where you are able to see like how the NCBI look like. So here you can see all resources and everything. You can, if you click, it will go to take you to the next page where you can able to read more, and it will redirect to toward the other resources, right? These are the popular resources. BLAST is the world famous software which is widely used. Similarly, like Bookshelf for searching gene, genome, nucleotide. Okay, so this picture is only depict like the front page of the NCBI, this PubMed database I made. So if you go to PubMed and if you do any, let's say if I'm interested in let's say coconut and disease gene. So I want to search articles which are on coconut and focus on disease gene. So the you can use boolean expression, like you type coconut and in caps disease, and if you search, it will give all the literature which contain 2 words, coconut and disease. So by this way you will get you will access this database, and it is updating regularly every day. Same with the evolution PubMed. Now you can search, and from here you can, for you, if you click here, it will be it will take you to the article, and you can save it as a PDF file format. The other databases like Kyoto Encyclopedia of Genes and Genome, which basically contain the information on genomics and its pathway, like on which pathway these genes are involved. There are a number of other databases which I'm not going to talk about, but yes, I'm going to tell you like this very important resource, or it's one of the first resource, which is a model data resource for the dicot plant, which is called Arabidopsis Information Resource, which is called TAIR. Okay, it maintains the genetic and molecular data about the higher plant Arabidopsis thaliana, so which is a model plant. Okay, it includes everything. It contains complete genome, its gene structure, its what the gene perform function, in which metabolic pathway it involved, and how the gene expression expressed. Okay, and whether, and along with its physical and genetic map, you can see the marker position. Plus, it contain the publication related to Arabidopsis also. So anyone, let's say if you are dealing with a new dicot plant, right, and but you couldn't able to, and you want to you want to amplify a gene which is which you think is it's almost common in all the dicot. So the best sequence or best primer pair you can design is from the Arabidopsis genome, because it's a model plan for the dicots, right? So, and the database is updated every 2 weeks. The genome sequencing is an endless process. The finishing takes time, or years and years. Now, that is all about the databases. So databases in terms like it's a collection, structured collection of related data in a structured format, so that you can user can make a query and extract the useful information and make it used for the further research. And the several databases combine to form a data bank. Now, I told earlier I told like the databases, the data is so in bioinformatics, the second important thing, the first one is like data, and second important thing is the format of data, right? So like let's say in the image processing, only the data format is like JPG or any image. In the protein, like in the mass spec data. But in bioinformatics, there are many data set. Okay, so like FASTA, everyone is aware about the FASTA file. It's a FASTA file format. It's a text file format, right? Which start with the greater than header, greater than sign, which is called header, followed by name. Name means it contains its detailed information about this below sequence. Okay, from where it was generated, what its function, whether it's a gene or anything else, right? Now this latest one is FASTQ. So these days, this genome sequencing, I know everyone is aware about the genome sequencing due to this pandemic. So the data coming from the Illumina machine is a major format is FASTQ format. By any machine, whether it's a second generation or third generation, the default is FASTQ. So what is that? It contains this ATGC information along with each base value. Okay, whether this base which is readed by a machine is of high quality or not. So that if it is a poor quality, the person who is analyzing, he has to remove this before processing further. For the protein like PIR, very common for the protein, MSF. So it's a sequence aligned format. So there are n number of format in data. So initially the data is in raw flat file format, like normal text file, which was stored in 1986 in GenBank and EMBL. But as data type is increasing, so they have changed the format, and from flat file, it has translated into 7 or 8 different formats. So like if this is a FASTA file format, so greater than sign by unique sequence ID. So this is an example of a human chromosome. So it is group 10. So this is GI information, and ML 4.5, some gene is there, and it is from the whole genome shotgun sequence. So by this way, the FASTA file format, but the FASTQ file format, it doesn't contain greater than sign. It always contain with a @ followed by the base and followed by the base quality. So this alphanumeric character what you are seeing here, it is an ASCII character. So all the computer science student who are available here, or for a normal student, the ASCII character are the character which are not understand or read by the human, but it is easily understand by the computer. Okay, so it will tell the base quality, like whether it's a very high quality base or not. Or let's say if initial base is low, so they have to remove it. Okay, and it can hold hundreds of millions of record in one single file. And the FASTQ file could be very large. It ranging from few MBs up to like 2-3 TBs or more than that. Now coming back to the another format, which is a GenBank file format, which everyone is seeing when you are accessing the NCBI. So this is the GenBank file format, and if I segregate this GenBank file format, so it structured in three basically pieces. One is header, second one is feature table, third one is sequence. The header itself, it only gives the information about this from where it was generated, what is the size, and who has decoded this one, is there any author. While NCBI and this feature table contain the, let's say if that's a sequence, so it's a CDS which starts from 77 and ended like 8814. The gene name is PNRQ, and it's a plant protein, right? So all this like feature table, and also it gives the translated form of that gene, and below the gene sequence information. So this is an NCBI GenBank file format. So I've already discuss you with, but this is again on a like on a zoom view. You can see. So this is feature identification table feature like gene. Gene is 1 to 15 KB, right? But the CDS starts from 79 to 969, and the location is like you see this locus is Aureus sativa there, and its function is like this is a putative carnitine protein, and its translated sequence. So by this way, the EMBL format also the same file format, but it end with with this termination line. So this is an EMBL file format. It's also header. You can see header, feature table, and sequence. Now this one is Swiss-prot file format. So it is a PDB file. You can see that this is how the PDB file look like. Okay, tell about this how many chain it is, like this the chain or like the helix where at what amino acid location is there, and then it will give you the amino acid sequence. Multiple sequence alignment, it was generated by the aligning the two or more sequences, and the format, when it is highly useful to find out the conserved and non-conserved region when you have a set of similar sequences. And so let's say if I am a gene, I'm having a gene which is related to the aroma of basmati rice, or let's say I'll take another example. So I have a gene which is related to the mango peel color, okay, across I have sequenced it from 50 different mango variety, including number Amrapali, like Mangnapalli, another. Okay, if I align those things somehow, like the peel color varies from among them 50. Okay, so that variation is due to the SNP in the or the nucleotide polymorphism in the at the sequence level. So multiple sequence alignment is always help us to find out the conserved and the non-conserved region in a group of similar sequence. Here you can see after alignment the block is telling like, okay, there is a mismatch, or you can say this red one is the similar block. You can see all the amino acids are similar, but in here you see GGDS, now KSSS, HAAT, and TTE, and but below before and after it is similar. So it helps us to identify this similar the conservedness in the at the domain level. Okay, and which will tell us whether this domain whether this SNP or mutational change is a main cause of that phenotypic expression or not. And also it will help us for the generation of phylogenetic tree. And okay, so the backbone of bioinformatics is what? Sorry, I'm hello. Oh, probably hello. Is I'm on time, or can I take 10 more minutes?
Yeah, we have enough time.
Okay, sure. Actually, I've become too
There is
discussed.
an answer in the data chat box.
Okay. Fine. Okay, thank you. Now, so can anybody tell like what is the backbone of bioinformatics? So the backbone of bioinformatics is the sequence similarity. If you see everything which is, or like just broaden your just horizon. Don't take this as a science, right? Or a bioinformatics. So let's say when you are searching a Google, right, any keyword, what it will do? It will find for this similar keyword, right, and put forth the result in front of you, right? So that is called similarity search. Similarly, the backbone in the bioinformatics, as we are dealing with the biological sequences, whether it's a protein or the nucleotide sequences, the backbone here is also sequence similarity. Okay, and the sequence similarity is based on the alignment. When you are, so this is an example. So alignments is what? A match of similarity between character, two or more sequence. Example, here you can see this is sequence 1 and this is sequence 2. Now when I align, so TT is a common, TA is not common, AGCATT, it is uncommon. So there are so many ways to align the above two sequences. The first way, like TT, TA, so the way it was written here. The second way, if I align like this TT, AA, so earlier only one base is aligning, now the two bases are getting aligned, right? Or in third way, where you can see at the end like these three bases are aligned. Okay, so which one do you choose and is the best? Can anybody tell 1, 2, or 3? Student?
There is a bit of a mismatch in everything. Maybe something with a gap would do better.
No, my question is like 1, 2, 3. You have an alignment. So which one, as per here, you will see as a selector? So if I were there at your place, I will select the third one. Why? Because there the three bases are matching. Rest is here two bases are matching. Here only one base are matching. Okay. So I will take only the best match or the match which is having higher base pair sequence similarity. Initially, in the before the discovery of that BLAST algorithm, the people are using the dot plots, for making this alignment and to perform the evolution analysis. So in this example, let's say this is an alignment of a protein. So HLTP EEEKSVHT and HAKSPEEKSVAT. These two sequences, let's say I want to align. So what they are doing, so they will make a dot plot. So like HH, so it's alignment, they will put across. Now L and A is not aligning, leave it. Now K and T not aligning, leave it blank. Now this K is anywhere else it is aligning, yes, here it is aligning, put across. Anywhere else, no. Same with P. P is aligning with P, but at not any other position. And with E is here also and here also. So first, by this way, they will put this X mark, right? And then they will find an optimal path where they are able to find out maximum number of similarity. So HH, now PEEKS, okay? PEEKS. So this PEEKS is a common. So they have selected this one, followed by the two mismatches, and at the end T. So earlier, so the blank region is a gap, while this line is a match. So by this way, they are doing this similarity search. But after the BLAST, right? Maybe some. But after the discovery of BLAST, it become automated, and this alignment become very faster, not manual. Now the now talking about the application area. So there are many application area like genomics, proteomics, system biology, and phenotyping. So if I say genomics, so decoding of the genome, or the prediction and iteration of genomic feature, their marker discovery, expression pattern, okay, sequencing, data analysis, which include gene finding and everything. If the proteomics, like protein, these is bioinformatics used for the 3D structure modeling, which is widely and heavily used in this pandemic for the designing and development of a new drug and vaccine. Okay, the system biology like gene set animated pathway and network analysis. This is also widely used, this network analysis, to study how any expression or the genetic expression or change in gene expression affect the whole thing. Now phenotypic is used for the GWAS analysis. So it's an association analysis. So let's say we have we have a number of phenotypic data, we can relate it into their genotype variation, uh, and can able to see this association analysis. So GWAS is the genome wide association analysis, which help us to identify the disease in case of human. If you're studying disease, it help you to identify like what are the SNP in which gene which that causes that disease. Okay, then like phylogenetic, you all are aware, na? It's basically the study of evolution relationship, right? So it's so this is an example of phylogenetic tree, right, in which it start with a root, right? So the root where the only ancestor is started. Now from root you see these branches which call the nodes. Okay, and from most recent, and the last one is the most recent ancestor. So by this way, in this picture you have 3 1 2 3. So these 5 sequence are like do AB are very similar, and they are from the same ancestor. The C and D are origin from the same ancestor, but AB and CD are like distantly separated, but their their origin is here only. So they are originated from the single sequence. So following tree are the thing which reflect the evolution relationship through picture or graph, and the rooted tree. So this is the rooted tree. So it will tell whether the ancestor is present or not. So there are another type of tree called unrooted tree, where you are not able to find out the ancestor. Immunopharyngeal area is its own independent area of research. So, and it is also a very vast field, which mostly depend on the computational biology or bioinformatics, and using that in this you can able to find, even you can able to find out like how many millions year ago this separation has happened. So this phylogenetic is very important. So just finishing this off. So I just want to just clear the doubt or the myth about 5 or 6 myth about what everybody knows. So myth number 1, okay, anybody can do it, right? So the most bioinformatician analysis can people think that okay, it can be done only you go to Google, open the web page, feed the sequence, right, and get the result. No, but that is not a case, it is not easy. So every question is a different user different approach to answer it. So the wet lab similarly, the person who was standardizing the same PCR for 3 different variety, same thing a bioinformatician is doing on the computer by changing the parameters to get the optimal or the best setting for that particular analysis, right? Okay, so because anyone cannot understand the algorithm, right? A bioinformatician can understand the algorithm very well, and based on that they can tune the parameters. If you are aware, if you run any software, whether it's a web based, it will ask for the parameter. So most 99.9% of the user, they are just running it on a default mode. But many times, it is not advisable to just rely on only one standard parameter. You have to play around with it, and then it will give you the best result. And people think, okay, and sometimes the problem comes where no software is available. So in that case, the bioinformatician or they will write new algorithms and scripts to solve that task. The second thing is bioinformatics is a service. Now the people and that is the biggest myth. Everybody know, okay, we generate data and uh, we generate data and we just give the analysis as service. This bioinformatics cannot be a service, right? Because it is a very important part of the research, right? So because it has to be go through very assumption, right? This so let's say the traditional biology, like hypothesis to evolution. Now using this high throughput biology, biologist can want to outsource this data to this bioinformatics or a bioinformatician, which is not true. But this bioinformatics is an interdisciplinary, like you can say, discipline, which require like the a team effort from statistician to a mathematician to a computer science for designing the better and the best parameter for their analysis. And most of the people who are sending their data to the company, they have their only one setup pipeline. They will simple do this, run your data in a pipeline, give you result. They don't care about like what is your assumption, what a user want. They just like give the data, what result comes. Okay, so it's a simple like garbage in and garbage out. So I know like most of the computer like students, they're aware like gigo. So this is garbage in and garbage out. If you're not understanding the problem, so the serviceman cannot deliver the service. So in this way, it is not a service, and it is everybody think, okay, bioinformatics is very quick, just on the click of button you will get the result. No, 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 this is not the quick thing, right? So because it require like a proper scientific experiment, designing, planning, and then the testing, okay, and the tuning the parameter, then finaling, analysis, and summarization of result. So it is not very quick. Even though if you are performing blast, you can change parameter and you can see the change in a blast. I will bet you like if you take like 20 base pair DNA sequence and perform a blast, it normal NCA blast, you're not able to find any hit. But playing the parameters, just if you change the word length from 11 default 11 to default 2, you are able to get the hit. Okay, so by this way, so it is not quick. You have to apply your mind. And yes, bioinformatician, they are doing it, even though they are long sitting, but they are analyzing the parameters. So the things to remember, right? The bioinformatics require a dedication and continuity, right? Like the way the wet lab person is required, the bioinformatics data analysis is full of research experiment itself, right? And most of our research, we work as an interdisciplinary research, and it is a team throughput. So by this way, I would like to thank you all for the kind attention and patience to listen this talk. And this floor is now open to take question.
Thank you so much, Shweta. Students, if you have any doubts or clarifications, you can ask. There is a question in the chat box.
So this is a chat, right? In chat?
Yeah, in chat.
So the question is, sir, do you feel a need for a centralized file format for bioinformatics rather than keeping track 20 of them like FASTA, BAM, FASTQ? Okay, Anjali, yeah, I think it is not possible, right, to keep a single file format, because initially it was started in the data population started in a flat file format, but it is not able to achieve that level, right? Because these is due to this technology advancement now, there are the data throughput is very high. So earlier the data is coming like a few kbs or mbs, but these days the data is coming in terms of gbs and terabytes. Okay, so this file format like the BAM FASTQ file format is a must need of this era, because like FASTA file, it only contain the base only the base information, but it is not containing the quality of base, right? But the BAM or FASTQ or SAM file, it contains the base information as well as its quality information. So probably you are going to see more file format, or might be some lesser more file format Like these are 20 or 19, it may be crossed 25 to 30 also. Because amplicon sequencing, there are one news I just want to update all the student, right? And for all the faculty, right? So we are earlier, we know like Illumina is used for the short read sequencing, and the PacBio or the nanopore is for long read sequencing. Now the technology is so advanced now. The Illumina is coming up with a longer read sequencing like the PacBio, and the PacBio is coming with a shorter read sequencing. Okay, so there is, and but their file format is totally different. Okay, and so by this way it's a technology changing. Probably this file format uh, will be increased. But yes, your suggestion is very good. Like if it is in less file format, it is easily uh, readable, and the data processing for the bioinformatician.
There is a question from Archit.
I hope, Anjali, I have answered your question. Okay, the one question is like from Archit. I would like to ask you a question as an individual who is entering the field. How one should learning the algorithm and language such as Python and R to manipulate the data? Archit, now actually learning, so there are two questions, like how one should learn the algorithm and language, right? So algorithm is nothing. Okay, algorithm is a way to solve a problem, or you can say flow chart, right? The flow chart is, so once you design a flow chart, you code that flow chart, right, into some language like C or Python to make an algorithm. Okay, and in that flow chart you are just keeping like, if this condition is fulfilled or greater than this, then it will go to that this way. If not, it will go to this way. So you first design a flow chart, then then you convert into algorithm based on the language, right? And the Python and R, Python is the easiest language to learn, and it is the future. You know, in here Hyderabad, it is mandatory to have Python, to learn Python, whether any student is taking admission at the university for the BSc, BA, or BCom, because so it is getting mandatory over here. So Python is very much easy, just like this the language we are speaking, like Hindi or Tamil, Telugu, or English we are speaking. It's a similar way, but just you have to uh, you have to just reread it, right? And a layman, even a 8th class student can able to code in Python. There are a huge number of resources which is available. Okay, and and it's a full of YouTube is full of this thing also. But anyway, if you like to know more about it, just be in touch with me, or you can visit my page where you can get my email address, and I will guide you to learn all those things, because I am going to organize the online courses also at my laboratory on Python and R for the student. So, and it is free and it is non-chargeable. One new message also come. Okay, fine. Anything else?
Any more questions?
I think no more questions.
Anyone else would like to ask?
Sir,
Yes, Kavi.
do you know of any sort of algorithm or something which will predict synergy? I have done some work on systems biology, but as far as I know, I've never been able to find something that will predict synergy between two ligands in dealing with targets. Is there something you know of that sort?
Actually, I do not much work on the proteomics part, but yes, there are tools like you can see, have you tried like Charm, or have you tried like this Charm algorithm?
No, sir. I haven't.
Okay, so there are in molecular dynamic simulation, if I talk about, let's say I want to see the probability of a ligand to bind a protein, okay, and I want to simulate it, okay. So the GROMAX is an algorithm which is widely used. It will tell you about the energy of your ligand, like at which position it is highly stable, okay, because a ligand which need to be bind with the protein, it should have a minimum energy, right?
Sir, I asked about synergy, not energy.
Oh, you want to know about synergy about ligand?
Yes, sir. Several drugs or ligands against a target.
Okay, that I don't know much, but if you drop your email, I will share that information with you. I'm more than happy to share with you.
Sure, sir. Thank you.
Thank you.
Kadambari, tomorrow we have a session for in silico drug discovery. Maybe you can ask that
Yeah.
the same
Sure,
question
sir.
tomorrow. You will get your answers.
Sure, sir. Thank you.
Any other questions? No more questions, then I would like to thank Dr. Ajay. Thank you so much for giving us such an elaborate lecture. Thank you once again from Srishti and Barrett.
Thank you so much, doctor, and for providing me this opportunity, so that I hope like I have encouraged student to take bioinformatics as a career, because it is going to be future. Okay, I once again I will tell you, like, everything is going to be automated, right? Not in the era of like 90s and 80s, where the people cloning is a big job, like people are doing, or sharing a DNA is a big job. Now it's obsolete now. Okay, the sequencing you have to just put. So let's say if I nanopore, in the nanopore you connect to your laptop, put a drop of DNA, and it will start doing sequencing. But the major drawback, or you can say the supply and the availability, you can the major scarcity is a data analyst, or a person who can understand biology plus IT, right? And there are huge job opportunities which is coming up, from Reliance to Tata to TCS, Google Genomics. Like now Amazon is also coming into this picture with Amazon Genomics. So this field is becoming more and more, like you can see, a hot place, and the job opportunity is more as compared to other field. Like you could be a database developer, you can be a data scientist, right? You could be a software programmer, which is for the genomics, right? And I know in Hyderabad only there are 50 to 60 companies which are MNC, and they are hiring at a very high rapid rate, and a very high scale also. So I hope like student can take this also in their mind, rather than fascinating themselves, because future is going to be digital. Always remember, everything is going to be digital, and digital is the future.
Thank you.
Okay, thank you so much, and bye bye. Thank you.